don't get super excited. Okay, this meeting is being recorded. That's totally fine. And then we're going to talk about beer on a chip, but literally just for one, and it's going to be a project that I've actually done with a bunch of students, so no graduate students involved, but we're a company, a local brewery, for beer on a chip, which is quite fun. Right, so what is a microfluidic chip? So the way I like to think about microfluidics or lab on a chip technologies, so if you think about you've been in a lab, or you're making a coffee or something like that, you're actually using your hands as the force that manipulates liquids, right? So you normally have, and you're pouring something together, something with your hands, the fact to manipulate glassware to do some kind of chemical reaction. You also use glassware, so that's a material that you're using. And normally you're working on that to me are relatively large, right? So you're working on the scale of several hundred milliliters or maybe tens of milliliters. But then if you think of chemistry from the biochemistry perspective, if you want to only say that when I'm talking to a chemistry audience, like biochemistry, you're going down orders of magnitude in terms of, right? So your reagents here are really pressure, working with enzymes or cells. So you now say a maximum of say a milliliter, but you're talking more on the level of a few hundred microliters. Also having to automate things to a certain extent because those volumes are so small. So you're using some kind of pipette to manipulate your reagents to be able to do an assay. And then effectively a different type of material as well, right? So you're using plastics because you care about contamination. You can do these assays and then throw them away and do the next assay in a completely new sterile environment. So in my mind, microfluidic technologies are kind of the next in this development of things to be able to do chemistry and biology. And so what we have here on this microfluidic device is we have, those are much smaller. So we're talking on the level of a few hundred micrometers, that's a maximum. So I'm just highlighting that there with my laser pointer, hopefully you can see that. So that's a channel on a microfluidic device. So what we're doing here is we are building a chip that is made out of some kind of plastic material, and we use those channels or pipes or valves or features on a chip to manipulate fluid, chemistry and biology on much scale. So that channel here that I'm highlighting is about the width of a human hair, so a few hundred micrometers, absolute maximum. And a host of advantages that I'll talk about in this lecture. And again, you're talking about different types of materials that we're using to do this chemistry and biology on these scales. Right, so none of that made sense. Let's imagine the imagination story here to see what, if you think about a chemical factoring plant of some type, like the pretty ugly one shown here, or you think about Charlie and the Chocolate Factory and the movie with Johnny Depp in it, and you think about all these tubes and pipes and vats, and I think even a waterfall, Charlie and the Chocolate Factory, using all these kind of pipes and things to mix things together to be able to make chocolate or chemistry or whatever. A chip. So in the chip, we would also have these valves and pipes and containers and use these to mix things together to do chemistry and biology on really small scales. So here is an image of a microfluidic chip that's quite big, and you can see here is not my image, but it shows you a couple of things here that is very important. So the science of small plumbing is what we're doing. So it's everything that has to do with plumbing, blockages, flow, how you pump things, that we have our microfluidic device. And if you are ever working in this area, dust will become your biggest enemy, because even tiny pieces of dust cause systems. But from a more scientific perspective, what we define as microfluidic technologies is that microfluidic technologies manipulate and process liquids and gases on scales below 500 micrometers. So gases is here in parentheses or brackets because we're not really going to talk about this in this talk, but we're talking about fluids as the general entity here. So this is a microfluidic chip. Here on the right is made out of PDMS, which is polydimethylsiloxane. It's basically stuff you put around your bathtub so that it doesn't leak. It's effectively the same chemistry there. And what you can see here is a bunch of channels in the middle. And I like this photo because it also shows you these big pieces of tubing that come into your chip. So here, somewhere off this image, you'll have some kind of pump that will pump these fluids onto your chips, onto your on-chip features, whatever you want on that chip. And the other thing to mention in this photograph here, so you've got a lighting system. So lighting is very important because you're doing microscopy, of course, a scale to be able to see what's going on in the chip. Now, if any of you have ever heard about microfluidics before, and perhaps gone to a lecture by someone else or something like this, we are not a community of research definition of microfluidic technologies are. So this definition is one we came up with in the paper that's cited below. And I'm going to talk a few slides. So there's lots of advantages. There's you know, you go to the scale for you when we publish. It's not just we can do this on a chip, but why? So obviously the first advantage, as I've kind of been showing you through the slides, is that we're using really tiny reagent volumes. So in this presentation, the reagent volumes that we're going to be using specifically when I talk about my research at the moment on bespoke artificial cells and tissues, we're talking on the nanoliter scale. So we're talking really on the cell scale of volumes. It's super high throughput. So I'm not going to talk about that too much today, but points of our technology is that we can do thousands of reactions per second, and we can analyze thousands of reactions. So this is a big part of what we do. We have this enhanced experimental control. So a bit in the similar way as there's that first, the round bottom flask to the pipette device. That miniaturization gives you much more control over fluids that you're working. Efficient heat transfer, that's a very it's the same process as, you know, you put a saucepan on the hob with loads of water and it takes 10 minutes to warm up, but you've got a tiny droplet on a microscope slide, you put a lighter under it, it heats up very fast. But this allows us to do some really interesting chemistry. So if you're interested in the more chemistry side and how we transfer this to kind of industry application, publication at the bottom will tell you a lot about this and why. And then small footprint, I should say that's a bit of a sarcastic one here. So we are chips are effectively the size of a stamp, which is really tiny. There's this lab around the chip that we don't really show. And the most are actually that microfluidic chip that had the tubes. But remember that you've got the chip, it's tiny, then you've got the tubes, and then you've got some pumping systems around you, you've got a microscope around you, you've got some kind of high-speed camera around you as well. And there's a lot of research out there on miniaturizing this, everything in one tiny chip at the moment. So this is here to remind you inversely that there is a lab that we work with. So my research is at the interface of chemistry and biochemistry. I'm actually a chemist by training. I only started doing microfluidics as a PhD student, but there is a heavy engineering component, and I'm not really going to today, given that the audience is mostly from the chemistry side. I've literally got, I think it's probably even 2 slides with a more engineering bent. So if engineering is not, you've got about a minute or 2 to see this slide, but it is a very simple background to the engineering. So why we care about what I'm slide is that we have to define what microfluidic experiment, right? So what, as I said, we don't really agree within the regime or how small we should be to gain all those hosts that have to do with microfluidic systems. So in the microfluidic realm, or when we're talking about fluid dynamics, we use a bunch of dimensionless numbers to describe fluid flow or characteristics or heat or things like that on these scales. And one of the most common ones that we use is the Reynolds number here, RE. And the Reynolds number looks at fluids and it talks about the forces in your system. And it's basically comparing inertial to viscous forces. If that doesn't make sense, don't worry. All we're looking at is what fluid are we pumping through our microfluidic device. We look at its density. We look at how fast it goes, we look at its viscosity, and then we've got this factor here, D, which is the characteristic dimension. So what is the dimension of your chip that has the most effect on fluid flow generally? And so what we did in this paper that I've cited down here is we tried to come up with a definition, design your device and what scale you should be working on to get microfluidic advantages. So we looked at loads of different dimensionless numbers. I'm just going to talk, and it's a very simple experiment. It's a very online experiment. It was actually a computational model that we developed a few years ago. So what we did here is we had a long piece of tubing 5 meters long, and we had where the interior diameter of that tubing was 5 millimeters, 1 millimeter, 500 micrometers and 100 micrometers. And then we pumped virtual water through it, and we looked at the residence time, so how long was that fluid spending, flow rate, and the gray box here on the slide highlights what the literature, what microfluidic researchers count as a Reynolds flow that has microfluidic properties. So this is where you would imagine the key advantages in terms of Reynolds flow happened. And so when you look at these 3 graphs, you basically see that to gain all the advantages from the microfluidic perspective, you really have to be working at really small scales. So your channels or your pipes on your device have to be 100 micrometers in diameter. Use much bigger microfluidic chips. And they are microfluidic, you're missing out on some of the advantages associated with that flow on the micro scale. So again, that's just an overview of kind of to show you the engineering side to what we do, care so much about fluid dynamics on this. Okay, so now we can forget the engineering for a little side. I'm going to talk about microfluidic droplets. So droplets is the area of microfluidics in which my group works the most. And here what I have at the top of this slide is a sketch of a chip. This is how we design our chips. We use AutoCAD to do so. And this is what we call a flow focusing junction. So I've zoomed into it here at the bottom and given you a video. So what's happening here is that from left to right, we are pumping in some kind of aqueous flow. And from top and bottom, we're pumping in an oil flow, and it pinches off that flow, that aqueous inner flow, to form these droplets. Okay, and we can make droplets in lots of different ways. This is just one of them. We've got these beautiful droplets, and from our perspective, each of these droplets is an individual reaction vessel. So theoretically, we can put whatever we want into each of these droplets, and we can analyze each of these droplets individually, and we can theoretically make these droplets at thousands per second. So that's like, imagine you're back in the lab, which I know you're not at UCL at the moment, but imagine you're back there and you're trying to do a reaction in organic chemistry. Well, we could do these at thousands per second, theoretically. So we're not going to talk about this in too much detail from the chemistry perspective, but more from the biochemistry perspective later on when we talk about the applications for my microfluidic device. So this is the typical video that you would see when we talk about droplet microfluidics. And so what is the reality, right? So I got relatively fed up with seeing these beautiful photos like this slide. I mean, these are the photos we see in papers all the time. They're the photos I've published in papers of our microfluidic droplets. And they're all spherical, right? They self-contain, they're not necessarily interacting with some volume. I mean, they even kind of glow on the sides if you look at them, right? And so I read papers and seeing these beautiful images, right? And I did really pick the worst possible photo I could get, but reality of what we see in the lab is not necessarily published. And I'm just going to zoom for you there to show how badly these droplets can go in the, I am exaggerating. So actually one of the first, the first paper I published by myself, and most of the work was done by Aaron. Aaron was an undergrad student in the Department of Chemistry at ETH Zurich at the time, and he did all of fantastic. And what we did is we got a microfluidic chip, we made drop a limit, and figured out when it broke effectively. So when the droplets changed from looking beautiful, contain and having the same volume to something a lot more disturbing from that perspective. And I love the chemistry project, and we'll talk a bit more about that in the next few slides. but it actually became a bit more biochemical, and I'll show you in a minute why, or biological, I think is the better word there. So what we ended up finding is that there were about 20 different or 24 different droplet failure modes. So when the droplets didn't behave like we wanted them to behave, and here are videos or images of all of those. I talk about this work and say it becomes more biological because what we did then is we got all of those droplet failure modes and we kind of built a family tree for it. So, you know, when you look at those biology diagrams of that fish and where it comes from and how it relates to all the other types and species of fish, this is kind of what we're doing here. So all those droplet failure modes that I showed you on the prior slides are here on the right-hand side. And then what we did is we categorized them depending on what physical or chemical effect was causing this droplet failure mode. So we've got two types. So either the droplets are in flow, so they're moving through your channels, or they're during storage. So we've stopped the flow when we want to, say, incubate them because we've got cells in them or something like that. So there are two types of experiments. And then we were looking at the underlying effect that it. So for example, if there's a problem with the shear forces or the interfacial tension, then you get certain types of failure modes, or you get wetting of the surface and surface affinity issues, and then you get different types of surface failure modes. Or you get surface failure modes that have to do with a surfactant. So surfactant is a stabilizing molecule. It's amphiphilic. It normally has a bit that loves water and a bit that loves oil, and we use them in our microfluidic droplets to stabilize our droplet systems. You don't have to, but I would say 90% of the time we do experiments, we use some kind of surfactant. And the reason for this is really clear. Like if you want to make droplets at home, you can just get a jar of oil and water, shake it up, you're making droplets, right? But as soon as you stop, so you remove that force that creates the droplets, those phases separate, right? Because they're immiscible. But when you add some kind of surfactant, some stabilizing molecule, then those droplets remain as droplets. And you see this all the time in real life. For example, when you make salad dressing, you've got your oil, your vinegar, you shake them up, you make droplets. But when you add something like a surfactant, say mayonnaise or like mustard or something like that, then that stabilizes that emulsion, and you're mixing, in inverted commas, the two phases. So of course, once you add that chemical to your system, then you have other effects on that system. You're not just working with oil and water. So this is a bit of theoretical chemistry, so I don't really do much theory, but this was a really interesting experiment. So what we did is we looked at our system. So this is a side view of our microfluidic channel here. We've got the wall at the top, the wall at the bottom. These are our three droplets. And we looked at where the surfactant was to end up within this droplet system. So we did some Langmuir kinetics effectively. You can look at this in more detail in the paper. And what we were looking at is as all these surfaces in the microfluidic channel as available sites for that surfactant to be, right? So for example, the total number of available sites includes the number of available sites on the wall surface, so your surfactant is sticking to the wall, the number of available sites for your surfactant to sit on the droplet surface, and of course, the number of available sites in the oil itself that is flowing this white thing around the droplets. And then you can look at distribution coefficients and the kinetics of all those surfactant interactions with different surfaces, and come up with some really interesting kinetics and graphs and things like that. And this was all done by Rob Witten, who's an author on this paper as well. And so I said, oh, this is fantastic, Rob. I really like what you've done. It's very interesting. I understand that, the chemistry, the kinetics of what's going on, but how does this help me in the lab, right? What does this mean realistically? So he plotted how many droplets we have in our system over how much surfactant is available for this droplet failure mode right at the top here, which we call dripping mode. So I've highlighted it here with a blue arrow, but I'm also using my pointer there to highlight it. So what's happening here is that your water phase is coming from the top, your oil phase is coming from the left, you're getting these lovely droplets, but the water phase is sticking here. So instead of shearing off at this T-junction corner, it's sticking and it's dripping off the end of this aqueous flow. So what we did here is we compared how this happens experimentally to how this happens as compared to Rob's model from this Langmuir kinetics over here. So the model is over here. We only have 4 data points. I know that's not fantastic, but the computation models took a long time to run. But I'm doing it here experimentally. So on both our x-axis, we're talking about water fraction. Water fraction means how many droplets do you have per volume of oil. So effectively, how many of all of this flow here, how much is water, how much is oil? And then here I was measuring from my experiment this relative length. So how bad was that dripping mode? Was it a tiny bit of dripping mode or was it a lot? And we get this kind of interesting relationship that adds that the more water, the more droplets you have in the system, the more likely you are to get a dripping mode here. And you see the same kind of effect from the model. And again, I said, okay, that's fantastic. Now I verified your model experimentally, Rob. So what? What does this mean if I'm in the lab and tearing my hair out because I'm not able to get this experiment to work from a microfluidic perspective? So he said, okay, we'll think about it from a different perspective. So what we're plotting here is how much surfactant is available, and we were a bit conservative. We said 90% is available instead of say 100%. And we also plotted, how many droplets we could simulate through our system. And what we see is that when we only have 2 droplets per second, which is really low in terms of microfluidics, our surfactant is effectively generally available, and you shouldn't be getting surfactant-caused failure modes. But as soon as you increase the amount of droplets in your system, so you go faster and you get more droplets, to 20 hertz even, which is really not very high, you're basically sucking that surfactant out of the system and creating chaos in terms of droplet failure modes. But you can flip this, right? So what we're saying here is that the more droplets you have, the less surfactant you have, the more likely you are to have a droplet failure mode. But theoretically, that means that if we take the droplets out of the system, we should be able to kind of reinvigorate our microfluidic device. So this is what I did. So at the top here, we have the same droplet failure mode, dripping mode that I showed before. You can see it sticking over here. And here I just took screenshots of that video. So on the left-hand side are the frames just before that droplet formed. And on the right-hand side are the frames just after the droplet formed. And from top to bottom is just basically a time series. So this is me starting the experiment. Right at the beginning, you can see how perfectly those droplets bud off just at that T-junction. There's no dripping, there's no sticking. And over time, this dripping mode happens. And you get this stickiness and you get droplets sticking and created from a dripping mode. But then because the model tells us that it's all about the droplets being there, we should be able to just remove the droplets, so stop the water flow in the droplets, and we should be able to regenerate those droplets back in there, and you get those beautiful droplets formed at the T-junction. Then over time, you get that sticking mode again as you have more droplets through that system, and you can repeat this process of going through that device recovery step. So that was pretty cool. Okay, so let's go into more practical aspects of chemistry. So up to what I've been telling you about is kind of the fundamentals of our droplet. I'm talking about surfactants, I'm talking about kinetics, but realistically what we do mostly in my research group is talk about, is use these for specific applications. So in this case, this was a collaboration with some researchers at DTU Denmark. And it was really interesting because they were able to injection mold their microfluidic devices. So you can see one there standing up on its slide. And one of the major failings, I guess, in a lot of the research we do in the microfluidics field is that we're using chip materials that are really not fantastic from the commercialization perspective. So you imagine trying to make a chip out of that silicone you put around your bathtub. You can imagine that it's pretty much like a sponge. I know you can't tell in your bathtub, but from a microfluidic perspective, everything gets sucked up into that PDMS device, and that's why we have all those a bunch of those droplet failure modes that we were talking about before. But also, you can't commercialize that. How do you even make that? How do you mass produce chips from that material? So one of the really cool materials that we like using are fluorinated polymers. This was a cycling olefin copolymer. In this case, it wasn't Teflon based, but we can also injection mold them, which means that you can make enough to be able to sell this. So what we wanted to do in this paper was look at why we would want to make the chips from these materials, what advantages did it have, and what kind of cool chemistry could we do on this microfluidic device. So here we've got, again, a flow focusing junction where we're making these little blue droplets, either the same scales that I was working on before. But we also have this huge feature on this chip. Well, huge for us, it's a couple of centimeters, but for us this is massive. Where what we were doing is we were making water and oil droplets, And then we were using gravity to separate them out. So we plug them all into this massive chamber, we allow flow to settle. We were able to separate out the aqueous droplets from the oil droplets for analysis, which is pretty cool. So just a reminder of the chemistry of this. So this is, it's a measurement that you use to look at whether a drug is going to go into a cell, right? It's a very, very rough and ready measurement. And the traditional way of doing this is you have a separating funnel and you've got a water phase and you've got some kind of oil phase, which is normally octanol or something like that. You add your drug to that mixture, you shake it up, you wait for a while and the phases separate out, and then you figure out how much of your drug is in your water phase and your oil phase, and that gives you an indication as to whether that drug is going to be able to cross the cell membrane. So I did exaggerate here, I did pick the biggest separating funnel I could find, that's 1 liter. You can get them obviously a lot smaller, and now you can do types of measurements computationally. But here what we were doing is trying to do this from a microfluidic perspective and figure out the advantages associated with that. So we did a couple of things. One of the things is more of the engineering side. So this graph on the left-hand side, what we were looking at is, well, how well does our device function, right? So this is a commercial system, so we need to, well, a potentially commercial system, so we need to kind of figure out at what flow rates we should be working, what droplet diameters we get, how good the separation between those phases. And this is kind of all this data here. And I'm not going to go into that in too much detail, but it's giving us effectively the parameters under which we should be operating this device. So we're really quantifying device performance then. But here what we are doing from a chemistry perspective, I think, is really interesting. So we're measuring obviously the partition coefficient or the distribution coefficient of quinine in this case. So we're effectively doing this shake flask method, but on a microfluidic device, okay? So the black data values here are us doing those reactions in the separating funnel, so off chip so that we can compare. The purple data point, I think there's only one, yeah, there is one, is the only data value in the literature that we could find so that we could add some data there. But these two violin plots here, blue and red, are the data values that we got from our microfluidic device. And we plotted them like this as violin plots instead of kind of averaging and adding an error bar or something like that, because it allows us to see how much data, so how many data values we get in one experiment. So the massive amount of data we just get from one experiment. And this just allows us to be a lot more robust in our statistics. And really understand. So this is kind of, I don't know, I think we probably did the shake flask method 3 times for each experiment. This is hundreds or thousands of measurements for one data value, and it just happens in an hour on a chip. So let's quantify that. So we're looking at traditional methods, which is your shake flask method, and we're looking at the millifluidic method. We call it millifluidic because it was a bit bigger from the perspective of a microfluidic chip. So if you want to do the shake flask method, you're talking about 24 hours to make sure that those phases are separated out. We get the exact same data or more data per data point in 30 minutes. Volume, you really want to be working on around the 20 milliliter scale. We're working on 300 microliters. The cost obviously is a lot less because you have a lot less reagents that you're using. You do have to add in the chip cost, but again, it's really small because you're injection molding. And then the interesting one here are the last 3 in my mind. So one is user sensitivity. So obviously our method, there is no user sensitivity because you're putting it on a pump and it automatically puts that through your microfluidic device. Whereas of course, if you're doing this from a traditional perspective, you have to prepare the right amounts and we have this human error added. You're talking about things like glass cleanliness. I'm getting flashbacks now to being in a lab as an undergrad, and all these things really affect your experimental output. In both cases, the repeatability is good. We can't say that this, you know, if you're doing your technique well, your repeatability is good from the traditional method as well. But the really interesting thing that we can do here is we can effectively just turn it on, put a detector on at the end, and we can just do continuous measurements of this system over time. So this might not be so interesting in this specific way of doing distribution coefficient measurements, but it does highlight something that we can definitely do from a microfluidic perspective in general. So I hope this kind of gives you a background of what a microfluidic chip is, why we care, and how all these surfactants and systems and chemicals or reagents that we have in our chip interact with each other. Again, a nice example here of using different types of materials and for different applications as well. So here's another one, and here you're going to have to take all those things I just said into account. So here we're switching material again. So this is a glass chip. And we're switching the phases as well. So let's go back one step. So this is about lipid digestion kinetics. And what we're doing here was a collaboration with the Laboratory of Food Sciences at ETH Zurich. And it was a really cool lab. They were basically interested in microstructures of foods and how that affects satiety. So they had this idea that how lipids or oily or fatty pieces of food interacted with each other within your digestive system might affect how full you feel, which is interesting. I don't quite know how they got to that hypothesis, but they couldn't test this using bulk methods, of course, because you just basically have a huge vat of emulsion droplets and you can't really see how those droplets are interacting with each other. So what we did here is we built a microfluidic chip and we made these tiny little wells where we could trap droplets to look at them over time. And so there's a glass chip and we're flipping the phases because we're interested in the digestion of lipids, so something oily. So these droplets are oil-based. And then what we were doing is we were flowing from left to right enzymatic digestion conditions that you find in your pancreas, so gastric or pancreatic lipolysis or digestion conditions. So effectively we had an aqueous fluid that mimics these two digestion conditions that are found in humans. So we had the pH, the salt competition, and also different types of enzymes there to mimic how these droplets would fare in a human being. So we made these droplets fluorescent. This is what you can see on the right-hand side, so that we could look at two different types of scenarios. So the top one here shows just one well in which we have 5 lipid droplets. And then we look at it over time as we change from gastric to pancreatic lipolysis or digestion conditions, and we're looking at how these oil droplets are effectively digested or munched by these mimics of your digestion. And then we did the same type of experiment, but where we only trapping one droplet, and that allowed us to compare how digestion occurs dependent on whether these lipid droplets interact within your digestion. So this is the second promised engineering slide, and I'm literally going to be on this slide for 30 seconds, so don't fall too fast asleep. But one thing we really wanted to check can be seen at the bottom here. We needed to make sure that regardless of how many of these lipid droplets we had in our trap, they were being exposed to the same fluid conditions in terms of the lipolysis, right? So we needed to make sure that they're being exposed to the same amount of digestive juices from that perspective. So what we did was a PIV experiment, so particle image velocimetry. So what you do here is you get the chip and you flow through it any fluid. It's normally some kind of aqueous fluid, which you've seeded with these tiny fluorescent particles. And so you can see the trajectory of each of these particles. There's loads of hundreds and thousands of them in the flow. And then we've got clever software that allows us to see how fast the fluid flow is in each section of our chip. And when we have these droplets in there as well. And you can see that there's very little difference from that perspective. So again, that was almost like a control experiment from an engineering perspective. So now we know it's not about fluid flow, it's about what's in that digestion mixture. So we got a huge amount of data out of this, and I'm literally just going to show these two graphs. And what we were looking at is how these droplets fare. So what is the diameter of these droplets over time? So this first half here is gastric lipolysis conditions, and this second half here is pancreatic lipolysis conditions. And these black data points here are when you have more than one droplet in each trap, and these white data points here are when you just have one droplet in each trap. And what you can see is that the diameter stays relatively the same during gastric lipolysis, maybe decreases a bit, and then during pancreatic lipolysis you get some interesting things happening. And you can kind of flip this over and look at volume solubilized, so how much of this droplet is munched into your digestive fluids. And again, in gastric lipolysis and pancreatic lipolysis over time, we can do some interesting statistics. Again, all of that's in the paper if you're interested. But what you're effectively calculating is that the microstructure does affect how fast digestion happens, or digestion kinetics. And this is really interesting for two reasons. Because if what you're saying is dependent on how those droplets in your digestive system interact, they get digested faster or slower, then you could say, okay, well, when I'm designing food products, if I can ensure that the microstructure of my lipid systems is less prone to digestion, then you're going to feel fuller for longer after you have eaten that food product. And it also has an interesting side effect in drug delivery. So we use lipid-based systems in drug delivery quite often to try and sneak drug molecules into cells, for example. But now we know that how those drug molecule envelopes interact with each other might be affected by digestion. So you might want to take into account as well microstructures, not just the chemistry, but how those molecules interact with each other in terms of drug release as well. Okay, so now we're going to move on to the more of the stuff that we do in my group at the moment. So again, we're going to flip the phases again. So what we're talking about here again are aqueous droplets surrounded by an oil phase. And what we're doing here is we're building artificial cell membranes on a chip. We call them droplet interface bilayers, or DIBs. And I'm just going to explain quickly what this means up here. So we've got an aqueous droplet, we've got an oil phase on the outside, and our surfactant that we're adding in this case are phospholipids. So these are the molecules that make up the cell membrane. They have a head group that loves your water phase, so they amphiphilic, but you've also got your tail groups that love your oil phase. So when you add these phospholipids to your droplet system, eventually what happens is that they form this monolayer of phospholipids on the surface of your droplet. And then you can use your microfluidic device, as I'm showing down here, to bring these droplets together, and you get a section of a bilayer, so the cell membrane between two droplets. So a couple of things to note here. Obviously, these images at the top are not to scale. So this is what you're really seeing, and your molecules are obviously thousands of times smaller, but this is just kind of exaggerating. So we're talking about a few nanoliters of a droplet. So this is probably about 50 micrometers in diameter, okay? So I'm exaggerating the scale here. The droplet interface bilayer, or the cell membrane section effectively is forming here. You get that characteristic kind of glowing pattern there. That's very common when a DIB forms. And this wasn't, I didn't invent these systems. They were done prior to us in the literature with this phospholipid here that I'm showing the chemical structure there called D5PC. It is not a natural phospholipid, so it doesn't exist in mammals, but it's a very nice one to kind of stabilize your system. And then people sometimes also add membrane pores like alpha hemolysin into this membrane here to check how that affects membrane dynamics. But the real thing that you need to remember about this whole system is that effectively you've got this artificial cell membrane section here, so this bilayer, but you've got this donor and acceptor compartment on either side of this artificial cell membrane. So let me show you that on the next slide. So this is very old work that I did during my PhD, but it's super cool. I always have it here because two things actually. One, because it was the first time this was kind of done on a microfluidic chip, which was pretty cool. But two, look at that microfluidic chip. It's so simple. This is just a square glass capillary with some pieces of tubing stuck in. You can see the piece of tubing here and the droplets coming out. So somewhere off shot here, we have a little droplet robot that you can use to make droplets. This was the first time we used that droplet robot. Dr. New made it. He is one of the authors. He was super cool. And now he's commercialized that system, which is quite cool as a way to make droplets. But it's very simple what we're doing. What we're doing is we're making droplets in this type of pattern here. They've got a DIB or droplet interface bilayer in between each of those droplets. And then they alternate in composition. So this one has a fluorophore in it. This one's empty. This one has a fluorophore. This one has an empty droplet. So over time, what you can see in the fluorescent images on the right-hand side is how much of this fluorescent dye goes through that droplet interface bilayer into the droplet, the empty droplet next to it. So the basic question that we want to ask at the moment in my research is, can we use these systems to model real human systems, right? So can we, for example, build a DIB system that mimics the pathway, for example, that a drug takes in the human body? So can we figure out how much of the drug that you dose in droplet 1 goes into droplet 2 through this artificial cell membrane, and how much goes into droplet 3 through this artificial cell membrane? So this is me with my analytical chemistry hat on. So what we're thinking is, okay, we have drugs. It takes 10 to 15 years and $2.6 billion to develop one new drug. And then not all of them make it to market because they fail at the clinical trial stage, right? So you've got your chemist right at the beginning developing the drug molecules, and then 10 years later they decide that it doesn't work because you can't get them into cells, right? And I'm slightly exaggerating the process, obviously, but not by that much. So what we want to do is we want to develop methods to test those early candidates for drug discovery and figure out whether they have this potential to enter human cells. How do they interact with cells, and do they get in? By how much? What can we tweak to make them better? That type of thing. So just a pause here to remind you of the technology that we're working with. Okay, so if you go back to that first slide where I was talking about using your hands to manipulate fluids, to do chemical reactions, obviously you can't do that on a chip, right? So even very simple actions such as mixing two fluids together, you have to build very specific chip architectures to be able to do this. And this one here at the bottom of this video is us diluting droplets. So even those simple actions are very hard. So if you think about it, we have to make chips, we have to dose reagents, we have to maybe incubate them, merge them, split them, dilute them, manipulate them. So all of that engineering has to happen with the on-chip features and architectures that we build. So these are two videos from my colleague, Dr. New, who just is a genius at developing these types of architectures on a chip. So we spent a lot of time doing exactly this, building the microfluidic side to be able to do these types of drug discovery experiments that we're interested in. So this is one of the chips that we developed and published recently. So what we have here is 3 ways to make aqueous droplets droplet A, B, and C. And then we bring them together. And in the middle here, we have kind of these rails, which I've highlighted here on the right-hand side. And so what these rails do is that they pick up these droplets and they bring them together so that you can form DIBs between the droplets here. So you can see them here. So droplet A, B, and C, and then I'm highlighting those DIBs with arrows there. And so we're talking, this is about 100 micrometers in diameter for a droplet. This is the chip in action. And what we also developed was a method to be able to immediately stop the flow when we had the perfect assay point that we wanted to develop. So this is just on a loop series. So this is, again, this is me kind of being really realistic about what really happens in the lab when we're working with microfluidic devices. So I could have totally edited this video or picked the right part of this very long experiment that shows just all 3 of them happening at the same time, none of them, you know, jumping rails or whatever. But I don't like that type of thing. So I'm very realistic with what I do. So then that's why we developed this system to be able to stop it when we get that perfect assay happens. This is very typical. Microfluidic things not being super predictable, but let's work around that and make sure. So when we stop the flow, then we can do these kinetic experiments over time and see what's happening between those DIBs. So this is what we're doing, right? So what we're doing here is that we were looking at pharmacokinetic compartment models. So if you've ever heard of pharmacokinetic compartment models, effectively what you're doing is you're looking at the body and you're kind of mathematically putting it into compartments. It can be 1, 2, 3 compartments, and looking at the distribution of your drug. So you take it, where is your drug mathematically predicted to be in the body? So we developed what we call a new type of pharmacokinetic compartment model, where we are able to predict where that drug is going to distribute in this system. So this system is for an orally administered drug. So we have one droplet here that mimics the intestinal space, the intestine. We have then another droplet that mimics an enterocyte. So those are the cells that line your intestine. And then we have another droplet that mimics your blood. And by mimics here, what I mean is that the pH and salt composition of each of these droplets mimics these compartments in the body. And then your phospholipids here that make up your cell membranes that the drug has to cross before going from one compartment to the other are made out of PC and PE, which is the major components of cell membranes for enterocytes. And what we did is we didn't use a drug in this case. We just used a fluorophore so that we could see it easily, and then we could quantify, if we dose it into the intestine, how much goes into the enterocyte and how much goes into the drug. So this is what the system looks like on a chip. And so we can quantify, right? So it's really easy for us to quantify how much is that. So again, you've got your intestine here, A, your enterocyte, B, and your blood, C. And so the first one, you've got all your fluorophore concentrated in time 0 in A, because that's where we dosed it. And you can see that very rapidly it disappears out of A and obviously goes into B, which is increasing nicely here. And then you can see that very slowly your fluorophore goes from your intestinal cell into your blood. And you can see again the increase here into your blood droplet at the bottom there. And we could do a lot of data analysis with this. I'm just going to show you a couple here. There's more in the paper if you're interested. But you can see how much is absorbed from that intestinal droplet as a percentage, and you can look at flux, and you can look at apparent permeability and things like that. It was really exciting because when we crunched all that data, we could show that our system, our little droplet system with these artificial cells, was 3 times better at predicting whether this fluorophore would interact, how it would behave with rat cells than the industry standard. So that was really cool, and saying our system is a lot more predictive than the industry standard pamper. So again, all that information is in the paper if you're interested in looking at that in more detail. So realistically though, okay, we are doing something very simple. So here we have, this is a DIB, this is what we're doing in our system, but the cell membrane is much more complicated, and I've done a mock-up here on the right-hand side. So A, you've got the bilayer going all the way around the cell, and B, you've got a load of other things in there. So you've got cholesterol, which regulates membrane fluidity, and you've got different types of proteins in this system, and you've got protein pores. So we're simplifying, so we're looking at passive transport, but we want to work with these much more complex systems, and we want to build them from the bottom up, right? So if you have a cell, imagine you could get a cell, you could add a drug to it and see what happens. You will see something happen, but because it's such a complex entity, you don't know exactly how. So what we're doing is trying from the bottom up to build that complexity and see how each of these extra components affects drug transport. So that's what we're doing at the moment. So the first one we're doing, this is just a video. Let's see if it plays. No. Okay. What we're doing is building the blood-brain barrier on a chip. So if you're looking for a PhD position or master's position, we've got open positions on this. It's a project funded by a New Frontiers grant, which is really exciting. So check out my Twitter account. I'll give you the handle at the end for the information about this. And what we're building here, and again, all you'd see is these droplets come together. So nothing super exciting from that perspective, but it's super exciting from the perspective that we're making these with porcine brain lipid extracts. And so this is the first time that someone's been able to build these artificial cells with mammalian systems, which is really exciting. So we're going to be building the blood-brain barrier on a chip using our artificial cells and build these artificial tissues to look at how drugs behave when you have Alzheimer's disease. So check that out. And there's some research coming out in here soon in publications, and also we have some positions open as well. We're also doing, and I'm nearly done, is some much more, I guess, I mean, they are definitely artificial cells and tissues, but there's much more about the origin of life. So this is done in collaboration with Pierangelo Gobbo, who looks really cool in the top right-hand side of this slide. And he's a researcher at the University of Bristol in the UK. And what he's playing around with is looking at, you know, from evolutionary terms, when you had that chemical soup on the surface of the earth, how did that agglomerate into something that was lifelike, right? How did cells form? And does this tell us something that is interesting right now to understand behaviors in terms of degradation and disease or things like that, or in terms of biomaterials, right? So he built these, what they call prototissues. So they're tissue-like entities. So they have a collective behavior, which is shown over here, which is the difference between just a bunch of cells or a tissue, right? So they have collective behaviors. And he builds them out of these protocells. And again, all this information is in his paper at the bottom here. But what he was utterly unable to do, because he's using these bulk systems, is build these to meet any type of specification. It was almost random how they came together. So what my grad student, Katherine, is doing is she's built a microfluidic device where we can build these prototissues. So he has 2 types of prototissue, so protocells, A and B, and then we can encapsulate them into these protocells. But we can encapsulate them in whatever configuration that we like. So we can do 1, we can do 2, 3, 4, 5, 6, 7, 8, 9, 10. And the ratio from red to green is also whatever we decide. So we can calculate how well we can do this. And hopefully all these videos will work. Yes, we can show all these different types of bespoke prototissues on a chip. And so this paper, we're literally writing up this paper right now, so I can't show you all of the cool data we have, but we can show really interesting behaviors dependent on the composition of these artificial tissues. Okay, last slide on the research side. As promised, is beer on a chip. So this is a project that we did in collaboration with, or we are still doing in collaborations with Phillips Brewing and Malting Company, which is a local beer brewery. And Ewan was a researcher there, and he was really interested. So I didn't know this, and I'm going to just blame the fact that I don't really like beer, which has nothing to do with fact with anything. But hey, so beer is an emulsion, which I didn't know. So if you like those IPA beers, which have the more hoppy flavors, your hop essential oils are effectively little droplets of hops in your mostly aqueous beer. So you have this emulsion there. And what they were finding is that the flavor, the hoppy flavor, degrades over time. And they didn't know why. So this is Katie. Katie is one of our first undergraduate students working on this. There's been 4 undergraduate students, and we're now writing this up as a paper. And what she was doing, and that is allowed, she has beer in the lab, and that's allowed because she was putting beer on a chip and looking at the emulsions there and building emulsions from different hop flavors and trying to look at degradation kinetics for that. So that was really cool. So check out our social media, and then we'll tell you when that paper comes out. So that's it. Thank you so much for your time, for coming to my presentation. I'm looking forward to having some discussion with you from now onwards. And I just want to thank my graduate students who obviously did a lot of this work, some undergraduate students, and all my other collaborators who helped with the work shown today, and obviously funding bodies. And yes, we have social media. They kind of make us, not make us do that, but they ask us to be on social media and talk about what we're doing when we new young assistant profs. So we've got an Instagram account where we show beautiful things that happen in the lab and when experiments go wrong in the lab. And we also have a Twitter account, which you can see all our group updates. And that's it from my side.
It's really weird when there's no clapping or anything. Thank you. That was such an interesting talk. I'm blown away by the sort of creativity from the link between these droplets to, you know, what it's become, the kind of the pharmacokinetic model and everything. Yeah, it's super interesting. So I'm going to open the floor to questions. If I think I can just let you unmute yourself, so you can just unmute yourself or raise your hand or type a question in the chat if you don't want to put your mic on.
The first question is always the hardest. It's definitely.
It's true. I have a question. I'm wondering what material you're making the chips out of. You mentioned an olefin copolymer, and why did you choose that one?
So that was actually, unfortunately for me, because I'd love to make all my chips out of it. It was only for that one experiment with the distribution coefficient measurements, and mostly that was because, how do I put this? I think sometimes we just do what's been done before in the field, right? So a while back, I think it was in the it's early 90s. George Whitesides, who was kind of one of the forefathers of the field, for want of a better word, made a microfluidic chip out of PDMS, so this polydimethyl siloxane. And since then, we've kind of all just used the same thing. And it's great because it's super cheap, it's easy to make, you make it in kind of the facilities that normal universities have. It's see-through, you know, you can throw them away when they get, you know, a bit too manky, and that's fantastic. You can't commercialize it. So that experiment we did with the distribution coefficients, and we use the cyclic olefin copolymer. That was injection molded, and then that's a different kettle of fish, right? You have to have an injection molder. You have to have an injection molder with a special inset that allows you to make features at the size that you want, because normally they're making features much bigger. And so I am literally about to put in a grant to see if they'll give us an injection molder, but we'll have to see. So I think part of it is that PDMS is readily available. So that's what we used for 90% of my talk and mostly in our lab at the moment. But yeah, if you have any genius ideas, you know, definitely come my way, because we want new chip materials. But I think some of it's just the kind of the inertia in the field is that's what we use because it's easy. Yeah. Oh, there's a bunch of questions in the chat. Shall I answer those?
Yeah, can you? Yeah.
How do you decide on this surfactant? Oh, good question, Susan. Okay, so it really depends. So again, there's a bit of inertia in the field from this perspective. I think, so our field is really interesting. It's not really something that you get to study as an undergraduate student, because you know, it's not a normal chemistry component in the degree or whatever. So usually what we have is that the research groups that exist are a bunch of researchers that came to this as PhD students or postdocs or whatever. And so they come from engineering, chemistry, biology, all types of background, and that's fantastic. But what we rarely get is an organic chemist who is interested in developing new surfactants for us, because it's kind of a bit of a service job, right? So there are a couple of surfactants that are available to purchase. They're very expensive and they work relatively well, but we don't really know what's in them, right? So you're adding this kind of chemistry muck to your droplets, and you don't necessarily know how that's interacting with your system. So normally we would kind of buy one of those if we can afford them and use them, because they're so good at stabilizing these droplets. In our later work, where we're talking about those artificial cells, of course our surfactant is those phospholipids. And then what we do, I didn't talk about it much because I didn't have time, but what we do is we build those phospholipid formulations to match the phospholipids that you find in different cell types, which is quite exciting. And in fact, what we're doing some work on cancer at the moment, because that phospholipid composition changes during cancer, and effectively that affects, or we think we're looking at whether that affects, for example, how chemotherapy agents interact with your cells as you get cancer. So that, I think that answers your question, but if it doesn't, feel free to ask a next one. Okay, so Stephen says, is it feasible to use 3D printing to generate your chips? Yes, it definitely is, and that is something we are playing around with. The problem again with 3D printing is you can't quite go small enough. You can, but it's quite hard. And also you want your materials to be as transparent as possible because of the microscopy that you're doing. So again, if you have them, and you want them, in our case, to be hydrophobic because you've got that water and oil system. So finding a material which you can 3D print small enough and be transparent and be hydrophobic is relatively hard. There's some really cool profs in the US who are working on that at the moment, which is really exciting. So I'm looking forward to seeing what comes out of their labs and then buying the system. But I love 3D printing, and we use it for other stuff, which I didn't talk about today. But the problem with 3D printing, it sounds so quick, right? You know, it's like printing a piece of paper, you just click print, it happens. It's not. It takes a huge amount of time to make sure that 3D print is working. Then you have to go back and change the parameters. So it's not just click and play, if you see what I mean, apart from the materials side. So we're not developing that for this type of chips. But hopefully we will be doing it when these genius researchers in the US come up with something specifically for microfluidics. So again, I hope that answered your question, and if not, just pop a question in the chat. So Ahmad says, how do the cells compare to the droplets in terms of size? Oh yeah, okay, I did answer your question, Seema, thank you. I'd assume any difference would reflect in a difference in passive diffusion between the artificial, and actual cell. Exactly, so that's a brilliant question. And again, something I didn't really talk about. So Ahmad's talking about those droplet interface bilayers that we made, and how the size of that droplet interface bilayers compares to the size of the cells that we're kind of mimicking. So in the case of that study that I was showing you with the intestinal orally administered drug system, our DIBs, our cells were about, I think they were 0.5 nanoliter in size. And I think the enterocyte cells, all in the paper, and I'm trying to remember the numbers, I think they were smaller. So enterocytes are smaller, but they're within the same order of magnitude, effectively. So we within mammalian cell order of magnitude, but there's, mammalian cells that are much smaller and much bigger. We're kind of in the middle, and the enterocytes are a tiny bit smaller. So it would have an effect, I think, in passive diffusion, maybe, right? But what we did is we actually normalized the data with regards to the surface area of the artificial cell membrane, because obviously that has an effect as well, and we normalized it for another couple of parameters like droplet volume changing over time and things like that. And the interesting thing is actually, so the commercial system that they use to do this type of experiments at the moment is way larger. So if we're on the 0.5 nanoliter scale, they're talking on the, I'm trying to not exaggerate here because again, I don't remember the numbers, but they're talking on the 50 microliters to 100 microliters scale, right? So we're not there, and that's something we're working on, but we're much closer. We're super close to doing it. And it's kind of a bit of a technology issue. Like the smaller you go in terms of droplets, the harder it is to make them and manipulate them on a microfluidic device. So that's what we're playing with. I hope that answered your question, Ahmed.
I think Yvonne's got her hand up.
Awesome.
Yeah, thank you. First of all, thank you for your presentation. I thought it was brilliant. My question is, at the end, where you introduce the 2, 3, 4 prototissues, I believe are called, what can you introduce there? Do they act like organelles, and how they interact between each other?
Oh, that is a brilliant question. I wish I could show you that data, but because we were recording and putting it on YouTube, I was a bit more conservative. Normally I share quite a lot of unpublished data, but because it's going out there for everyone, I was a bit on the scared side. So yeah, we're doing some really cool stuff there. And we're actually doing some enzymatic chemistry inside the protocells. So one thing we actually had to do when writing the paper was define is what is a tissue, right? I'm not a biologist. So tissues, you know, some of the characteristics that define tissues, that they have collective behavior. So they have, they somehow have a behavior as a whole that isn't just a behavior of a couple of cells in a you know, sitting around in a buffer, right? There's this communication, and also they can communicate with each other. So we're doing a bit of both in that work. So we're doing collective behaviors in terms of thermoresponsivity. So the way that we designed the chemistry, or Pierangelo more accurately designed the chemistry, was that so we had these thermoresponsive components here, polymers. And so it's really cool how you're able to see these collective behaviors and how they correlate to how you've built that prototissue in terms of how many protocells and the ratio and the geometry that we have built from the bottom up, which is pretty cool. And then so actually the paper, the first paper, the Pierangelo paper on that first slide of the prototissues, was the first time that anyone had really built a prototissue. So this is really early work. And what we did as well is, in terms of communication of those prototissues, we're doing some kind of, I'm trying to not say too much, but trying to do some enzymatic reactions in them, where we do part A of the reaction in one protocell, and the reagent that is created sneaks into the next protocell and creates kind of an enzyme cascade type thing, so that we can show that they communicate, and again quantify that based on how we built it and the geometry. I think that answers your question in a not giving too much away way.
Thank you very much. I would definitely be checking the paper when it comes out.
Yeah, thanks, Yvonne. That's awesome. Yeah. I can't see, I think you're on mute. I can't see any hand raising. I don't know if that's because I'm not a host. So
no, I don't think there's any more hands up, and I think that's all. Oh, there's another
question.
Chat.
Susan says, would it be possible to attempt microelectrophoresis with some of the extras? What extras, Susan? You mean in the prototissues or in the, oh, sorry, you're on mute, Susan, so I can't hear you.
Sorry, just if you were adding some phospholipids, you'd have charges about. So I just wondered if you could count the droplets going forward and backwards. It may not be, because it's always a difficult subject. Just wondered.
I don't know. That's a really good question. Not something I've really thought about. So we definitely do electrophoresis on a chip. Well, not we. It's done electrophoresis on a chip. I haven't really thought about that, and I don't necessarily see quite why not. We are interested in charges from a different perspective, actually, because we've been finding some really weird effects. We've actually become biophysicists as well when we started working with phospholipids, because we've seen some quite strange effects on how these bilayers are built related to temperature and charge and saturation effect. So we're actually writing that up. So I'm going off on a tangent here. I'm not really talking about electrophoresis anymore, but I'm talking about charge in these systems. So people do use electrophoresis on a chip to move things around and do stuff, but we haven't, is I think my answer to that question. I think that's probably one of the hardest questions I've ever had. Thanks, Susan.
It seems so interdisciplinary. Like you really need sounds like you really need the engineers' help, and they need you to make what they're doing worthwhile. And yeah, it sounds like all really cool projects in their own kind of respective ways.
Yeah, it is. I mean, I really like it because I feel like you can just come up with something new and go, oh, okay, well, let me just pick expertise from here and here. But I think it is relatively stressful sometimes when you come into the group new, because there's so much to learn right from the start, and then it sounds like we all know so much of what we're talking about. We don't. We're just kind of quite broad. But we don't necessarily go into depth. Well, we do go into depth, but sometimes, as I said, the last paper that we're writing is about biophysics. I mean, that's insane. This is not my area of expertise, but you start looking at something and it goes a bit crazy from that side, so you have to do it. Oh, cool. Another question from Harry. Is the research currently solely focusing on droplet-based vessels and reactions and diffusion in between droplets? Have you attempted any organic synthesis drugs, maybe using microfluidic flow chemistry? Okay, so yes, at the moment, I am mostly working on droplet-based systems. Let me check that through. Yeah, I don't think we have any non-droplet systems at the moment in the lab. I've definitely done non-droplet systems before as a postdoc and things like that. And in fact, so is it only droplet based? Yes. And reactions and diffusion in between droplets? Yes. So we are going to take that a bit further as well. Sorry. And work with, I guess, go even more biological and try and add some protein pores to that, mammalian pores. So that should be exciting. Have we attempted any organic synthesis drugs? Maybe. Wait a sec. Sorry, talking to myself is hard work. So I have, actually as a postdoc, I did do some organic synthesis in a completely different system. We were doing some singlet oxygen chemistry. It wasn't for drug synthesis, but actually that paper I kept citing at the beginning as well was a perspective piece in Nature Chemistry, talking about microfluidic flow chemistry, because we did a big kind of perspective review on the field. Based on some kind of work we were doing at the time. So I haven't done any flow chemistry for drug synthesis. I've done flow chemistry for organic synthesis. And again, all of that's kind of older work, so not right now. Does that answer your question, Harry? I think so, but feel free to ask more.
Yeah, there's no one to sort of nod or, there we go, yes, thanks. Well, I'm going to hand over to Chiara now. So hopefully we can try and recreate, you can give your voice a bit of a break. We can try and recreate our sort of after talk chats.
Sounds great.
I'll hand over to her. Thanks again.
You're welcome. Thank you so much. It was great to meet you.